There is a rule you were taught in school, even if you never noticed learning it. When an animal dies, the soft parts rot away. Only the hard parts last long enough to turn to stone. That is what a fossil is. A rock doing a very good impression of a bone, with nothing of the animal itself left inside. In 2005, a paleontologist named Mary Schweitzer dissolved the minerals out of a 68-million-year-old Tyrannosaurus rex bone from Montana. What should have been left was nothing. What was actually left was stretchy, soft, flexible, see-through, branching vessels that bent when she pushed on them and sprang back into shape. Little round structures that looked under the microscope like cells. All of it sitting inside a bone that had turned to solid rock long before the first human ever walked. This was not a fresh kill. This was one of the most studied predators in the history of the planet, dug out of solid stone, sitting in a museum collection. And the tissue inside it was behaving like the animal had died last year. Here is the problem that started the moment she published. Everything known about how fossils form said this could not happen. Soft tissue does not survive 68 million years. It does not survive 68,000. The textbook number for how long protein could last before it broke down completely was about 1 million years. She was holding something roughly 68 times past its own expiration date. So a lot of scientists did not call it a discovery. They called it a mistake. And the argument that started that day is in some ways still going right now. By the end of this, you are going to have a hard time looking at a museum fossil the same way again. Start with why nobody had seen this before, because the answer is a little uncomfortable. To find soft tissue inside a fossil, you have to destroy part of the fossil to look. You take something rare and irreplaceable, something a museum would insure for a fortune, and you dissolve a chunk of it in acid on the off chance that there is something soft hiding in the middle. For most of the history of paleontology, no serious scientist would do that, because everyone already knew what was inside: rock, just rock. Looking would be vandalism in the service of a stupid question. She looked anyway. And the reason she looked is almost funny. She had been studying the bone under a microscope, saw structures that looked like blood vessels, and could not unsee them. So she ran the test everyone assumed was pointless. What came out was a network of transparent, flexible vessels, a fibrous, stretchy matrix, and cell-like structures with tiny branching arms, the same shape as the bone cells in living animals. Some of the vessels even had small round objects inside them, sitting roughly where you would expect blood cells to be. There is one more detail about this exact bone that makes it stranger still. When the team looked closely, they found a special layer on the inside called medullary bone. That is the tissue living female birds grow to stockpile calcium when they are about to lay eggs, which means this individual was almost certainly female, and she died while carrying eggs. Researchers nicknamed the specimen B. rex. So the single most famous piece of dinosaur soft tissue on Earth came out of a pregnant female T. rex, found half by accident, by a scientist willing to break a fossil open to see what nobody else would look at. But something that looks like tissue still has to be proven to be tissue. The obvious counter-argument was that this was some kind of slime, a film of modern bacteria that had grown into the shape of vessels long after the dinosaur was gone. To answer that, you cannot just look harder, you have to find the actual molecules. So a second scientist got involved, John Asara, a mass spectrometry expert who normally hunted for proteins in cancer tissue, took a sample of the T. rex bone and ran it through one of the most sensitive instruments in the field. He was looking for collagen, the main protein in bone, and specifically for its sequence, the exact order of its building blocks. He found it. 7 fragments of genuine collagen protein pulled straight out of the T-Rex bone, their sequences confirmed independently by mass spectrometry. And then came the part that got everyone's attention. When he compared those dinosaur sequences to living animals, the closest match was not a lizard, it was not a crocodile, it was a chicken. Sit with that for a second. The most fearsome land predator that ever lived, the animal on every lunchbox and museum banner, and the closest surviving relative of its actual proteins is the thing in your fridge. A follow-up study a year later ran the T-Rex collagen against a whole roster of modern species and reached the same verdict. It grouped with birds, with ostrich and chicken, more tightly than with any reptile. It is worth being fair about the chicken result, because the scientists were. The match was partly a matter of who had already been sequenced. Crocodiles and alligators, the other obvious relatives, had not had their proteins read yet, so a chicken was simply the closest thing on the shelf to compare against. Even so, the finding held up. When more animals were added to the comparison later, the dinosaur still landed with the birds, closer to an ostrich than to any living reptile. Which is a genuinely strange thing to have to say about the tyrant lizard king. Scientists had suspected for a long time that birds descended from dinosaurs, but that idea was built almost entirely on the shape of bones. This was the first time anyone had reached into a dinosaur, pulled out the molecules and checked, and the molecules agreed, which should have settled things. It did the opposite. Because a claim this big gets tested hard, and the tests got brutal. Other researchers reanalyzed the data and pointed out that several of the dinosaur sequences were suspiciously close to ostrich, and that the same lab had handled ostrich samples. That opened the door to contamination. The idea that some of the so-called dinosaur protein was really modern bird that had wandered into the sample. One reanalysis even flagged a fragment that looked like bird hemoglobin. For years, the field split into 2 camps, and they did not split politely. One side saw genuine dinosaur molecules and the discovery of the century. The other side saw wishful thinking, contamination, and a story too good to be true. Every new paper got picked apart line by line. And underneath all the shouting was one honest, enormous question that nobody could answer. Even if the tissue was real, even if the protein really was dinosaur, how? What possible process keeps something as delicate as a blood vessel intact for tens of millions of years when that same tissue in a dead bird on the sidewalk turns to nothing in a week? For a long time, the honest answer was that nobody knew, and a discovery with no mechanism behind it is very hard to defend, so the team went looking for one. And what they found was, of all things, rust. The key turned out to be iron, specifically the iron packed into hemoglobin, the molecule that makes blood red and carries oxygen through the body. In a living animal, iron is dangerous. It is so reactive that your cells keep it locked up and escorted everywhere it goes, because loose iron throws off free radicals, aggressive little molecules that tear other molecules apart. It is part of why we age. But after death, that same destructive chemistry can flip into something else. When an animal dies and its blood cells break open, all that iron spills out. The free radicals it throws off can grab nearby proteins and weld them together, cross-linking them into a tougher, tangled mesh. In her words, it works a bit like formaldehyde, the chemical used to preserve bodies. The very thing that ages you while you're alive can, in a sense, embalm you once you are dead. To test it, the team ran an experiment that is almost comically simple. They took blood vessels from modern ostrich bone and split them into 2 groups. One group they soaked in a solution rich in hemoglobin. The other they left in plain water. The vessels in plain water rotted into mush in about 3 days. The vessels soaking in the iron-rich solution sat out on a lab bench at room temperature for more than 2 years and still looked intact. One ingredient made all the difference. And there was a second twist. The same iron that preserves the tissue also coats it and hides it from the tests scientists normally use, which helps explain the awkward little mystery of why nobody had noticed this stuff sitting inside fossils for the previous 200 years. Now, iron does not make the whole problem vanish, and this is where the story has to stay honest. Keeping a vessel intact for 2 years on a lab bench does not prove it can last across a stretch of time the human mind cannot really picture. Critics pointed that out immediately, and they were right to. What the mechanism did was turn the impossible into the merely improbable. That is real progress, but it is not the same as closing the case. What it did do was change the conversation. Preservation was now a chemical process with specific conditions attached, not an unexplained miracle. It seemed to need the right kind of burial, and fast. Something like dry sandstone that sealed the bone off from water and bacteria with plenty of iron on hand. Those conditions are demanding, which is exactly why preserved soft tissue is rare. But it should not be a one-time event. And if this is chemistry, then it should have happened more than once. Which brings us to the reason this old fight is suddenly back in the news. In 2025, a team led by researchers at the University of Liverpool went looking inside a completely different dinosaur. This time it was not a Tyrannosaurus rex at all, but an Edmontosaurus, a duck-billed plant-eater, its bone roughly 66 million years old. Using modern chemical analysis, they reported finding evidence of endogenous collagen, original protein, still inside the fossil. A separate group identified hydroxyproline, an amino acid that acts as a specific fingerprint of collagen and is very hard to explain away as contamination. That matters because it means the Tyrannosaurus rex was not a fluke or a one-off lab error. A different animal, found at a different time, studied by a different team, is telling the same impossible story. The soft stuff is in there, in more than one dinosaur, and the list of specimens showing it keeps getting longer. So where does that leave us? It leaves us with a genuinely open scientific fight, which is a rarer and more exciting thing than most documentaries will admit. The mechanism is real and testable. The molecules keep showing up, and yet the sheer scale of the time involved still makes some very smart people deeply uncomfortable, and they are not wrong to feel that way. That tension has not been resolved. It is being argued over, in real journals, right now. But step back from the fight, and the plain fact underneath it is staggering. There is a decent chance that inside certain fossils, sealed in stone, since before there were primates, before the flowers we know today, there are still fragments of the actual animal, not a mineral copy. The thing itself, protein that a living dinosaur made with its own body, and that has somehow been sitting in the dark ever since. For 200 years, the assumption was simple. A fossil is a stone portrait of an animal, with nothing of the animal itself left inside it. Some of them, it now seems, may have been quietly holding on to a trace of the real thing the whole time. So here is the question worth arguing about, and I would honestly like to know where you land. Can a lab experiment that ran for 2 years really tell us what happens to tissue over 66 million? The experts are split on exactly that line, so drop your answer in the comments. You are allowed to be split too. And the next time you are standing in a museum, looking up at some enormous skeleton with a confident little label bolted to the base, it is worth remembering. The bones might not be empty. Some of them are still holding on to a secret, and only in the last few years have they started very quietly to give it up.